Hello, and welcome to our lecture on transcription and translation. So we have covered DNA replication and how bacterial cells replicate their DNA prior to binary fission. Now we're going to look at the process of transcription and translation. Now in biology, we have something called the central dogma. And the central dogma, or what is central to life, is DNA makes RNA, makes proteins. And this process here from DNA to RNA is known as transcription. And from RNA to proteins is known as translation. And both of these processes have the same three steps. Those three steps are initiation, so what starts it, elongation, the process of actually making something, and termination, which is the end of the process. Now, how each of these three steps, initiation, elongation, and termination, occur is different between transcription and translation, but the idea is the same. So let's first start with how to actually read our genetic code. When transcription occurs, transcription is taking a DNA sequence and converting it or copying that DNA sequence, but using RNA nucleotides, because RNA nucleotides are what are going to be read by ribosomes for protein synthesis. And from DNA to RNA in transcription, we simply go from one nucleotide type to another. So we might have our DNA code. Right? Our DNA code would be T, T, C, G, A, C, C, T. And then when we go to our RNA code in transcription, we would go, instead of T, we would have an A. So it complementary binds. This would be a U because we're using U instead of T. Then A. So this is the RNA sequence of the gene found on the DNA. So the DNA itself is not actually interacting with ribosomes, the RNA transcript is. And we are changing the language from a deoxyribose sugar to a ribose sugar so that it is recognized by the ribosomes for protein synthesis, for translation. But when our proteins are made and when translation occurs, the DNA sequence is read one base at a time, but the RNA sequence is actually read 3 bases at a time. And these are called codons. Now, this table on the right is kind of part of this diagram, and this is how we used to read the codons. If you notice, this is in DNA language. So if the gene is TTT, then the RNA would be AAA. So this particular table on the right is used to look at codons within the DNA molecule itself, which is very confusing. So I'm going to cross that out over there, and I'm going to have you look at this wheel here. We're going to be reading DNA in terms of RNA sequence. We want to read the RNA transcript and look at what is actually going to be encoded for. And we have to translate this RNA sequence into a amino acid language. So we're going from RNA language, genetic language, to amino acids, which is a protein language. And so this translation occurs via ribosomes. And what's really happening is when ribosomes read, when they read that mRNA transcript, messenger RNA, they read it 3 bases at a time. So let's take a look at our transcript down here at the bottom right. I have the transcript AAG. So when AAG, this is referred to as a codon. I'm going to put my first codon here. If AAG is my first codon, this codon encodes for the amino acid. Well, here's the first amino acid. So this is 1. This is 2, and this is 3. So it encodes for the amino acid lysine. My next codon is CUG. See, 3 bases at a time. So if my next codon is CUG, then this is going to encode for 1, 2, 3, leucine. Then my next one is GA. Let's just add a U here just so we have it. So my next one is GAU. If I have GAU here, that's going to encode for 1, 2, 3 aspartanine right here. So this would be ASP. And in following this sequence of codons in the RNA, which is transcribed directly from DNA, I now have the primary sequence of a protein. And this will continue for every 3 bases. And I now have the primary sequence of a protein. So in protein folding, we go from primary to secondary to tertiary to quaternary. And my protein can now fold properly because I have the correct primary sequence or primary structure of a gene. So that's how we read the genetic code in terms of RNA. So over here, this is RNA. And over here on the right, I have the same thing being read as DNA. There we go, as DNA. So here we have just kind of an example of transcription and translation. I have transcription over here on the right. So this is transcription. Transcription is really very simple. It works a lot like DNA replication, but instead of DNA polymerase moving along the gene here, I have RNA polymerase. Right? RNA polymerase. So RNA polymerase started over here somewhere at the regulatory region where everything gathered, and then it starts reading, and we have this little bubble here. This is referred to as a transcription bubble. And the transcription is occurring on this one. We'll just say this is our 5' strand and this is our 3' strand. So in this one, RNA polymerase is moving to the right. And it's moving into the right because this 5' right gives me that 3' there. And my large long pink line here is my RNA transcript. Now this RNA transcript is currently in process of being made. And you notice here, attached to my transcript are ribosomes. And in bacteria, transcription and translation is a simultaneous process. And this is because we do not have separation of our DNA from ribosomes like we do in a eukaryotic cell. In eukaryotic cells, messenger RNA is transcribed in the nucleus. It goes through a process, some processing, and then it moves out into the cytoplasm, and translation is a second process. And they occur in spatially separate areas. So they occur independent of each other. Whereas in prokaryotic cells, transcription and translation are occurring at the same time because we do not have a membrane-bound nucleus, so that mRNA transcript becomes available as soon as it's being made. So I have multiple ribosomes all attached to this transcript here, and it's all moving through as RNA polymerase is still synthesizing this mRNA transcript. We'll look a little bit more into what initiates transcription, but the concept of transcription, initiation is done with what's called an initiation site. It's an initiation sequence. It's part of the regulatory. It's called a promoter. So that's initiation. Elongation is just synthesis of the mRNA transcript. And then termination is a termination sequence. Just a termination sequence. So transcription is a pretty simple process. Just remember that translation can occur at the same time as transcription. Now over here on the left, we have a ribosome that is in the process of translation. And translation is a little more in depth, right? There's a little bit more to this particular process. Now in translation, the first thing that's going to happen is the ribosome itself, the small subunit, this part right here, is going to attach to the mRNA molecule, the messenger RNA, and it's going to do this at the 3' end. It always attaches to the 3' end, so you can see at the 5' end, and it's going to move towards the 3' end. So it's looking for this 5' end. And at the 5' end is going to be just kind of a short, benign type sequence. There'll be a couple of, we'll have a couple of bases here. And then we will have a very specific sequence called the Shine-Delgarno. And in the Shine-Delgarno sequence, this is a very specific sequence that's conserved that's conserved in bacterial DNA. And then transcribed into the RNA. And the Shine-Dalgarno sequence is used to initiate. And in initiation here, the Shine-Dalgarno sequence sets what's referred to as the reading frame. Now, the reading frame, remember, we're reading this mRNA transcript 3 bases at a time. We're reading it as codons, which means we need to know where to begin. And in the codon table, or in the genetic sequence, is the start sequence, right? The start codon. There's only one start codon, and it is AUG. So what the Shine-Dalgarno sequence does is it raises a flag to the ribosome and says to the ribosome, once you see me, the very next AUG that you come across is the first codon of this protein. Now, AUG as a start codon also encodes for what's called FMET. FMET is formyl methionine, and that is the methionine that is used in bacterial proteins. Methionine is an amino acid. Now, this is a specific amino acid called formyl methionine. It is unique to bacteria, but it's still methionine. Now, that methionine may be processed off later, so not every single protein in bacteria has methionine as its first amino acid. It may get broken off, but it is the very first amino acid that is brought in to the ribosome to begin the elongation process. So in initiation, right, initiation of translation, we're going to hit that SD sequence, that Shine-Dalgarno sequence. And once we hit the Shine-Dalgarno sequence and the first AUG is found, we begin elongation. And elongation in ribosomes occurs in 3 places within the ribosome. And then, of course, at termination is when we would reach the end of the mRNA transcript, and the whole complex falls apart, releases this polypeptide out into the cytoplasm, and the cell is now has the protein that it needs. So here's a little bit better diagram for showing our process of transcription and translation. So you can see up here, this is showing it in eukaryotic cells, by the way. This would not be occurring in prokaryotes, but in eukaryotes, our mRNA is in the nucleus and then moves out into the cytoplasm. But I wanted to choose this because I wanted to show the ribosome process here. And what we have is we have tRNAs. tRNA stands for transfer RNA. We have our mRNA, that's what's occurring right here. And within the ribosome, there is actually what's called ribosomal RNA. And the ribosomal RNA is what actually recognizes the Shine-Dalgarno sequence. This is RNA that is part of the actual ribosome itself. So recognizes Shine-Dalgarno sequence. And that Shine-Dalgarno sequence, again, is kind of that big flag. It's the entry point. It says, okay, ribosome, get ready, because the very next AUG is the start codon, and you're going to have to start translating from AUG on. And when it hits that AUG, what happens is the ribosome now knows which 3 bases to start. And at that AUG, it then will now move to it will move 3 bases at a time. As it moves 3 bases at a time, over here we have what's called the A site of the ribosome, the P site, and the E site. Now in the A site, this right here is that's our codon, right? They're showing you here where the codon is. But notice on the tRNA, we have these 3 bases here. These are known as the anticodon. And what we're going to do is the ribosome is going to match the anticodon of a tRNA to the codon of the mRNA. And for all 20 essential amino acids, or any of the amino acids that are required for a cell's proteins, whatever combination of amino acids they have, each amino acid will have its own tRNA. So here's the anticodon that will match the codon, complement base pair the codon. And at the opposite end of the anticodon on the tRNA is the actual amino acid that it encodes for. So the ribosome is going to randomly throw these tRNAs into the A site. A stands for acetyl site. And they randomly throw these tRNAs into the acetyl site, and they will, once the correct tRNA that has the matching anticodon binds to the codon being presented, then the ribosome will go through a conformational change, and both the mRNA and the anticodon in their base pairing right here at the bottom in this base pairing will move over to the next binding site, the P site. The P site stands for peptidyl. And in the P site is where we hang on to this growing polypeptide. And what is happening is this amino acid here, we're going to call this 1 and we'll call this 2. Amino acid 1 is going to bind to amino acid 2, but this amino acid 2 is going to get attached to it. All of our tRNAs move over 1, the mRNA moves over 1, and this amino acid and tRNA will now be in position P, will be in the P site. This will expose our next codon that will be looking for the anticodon. And this particular amino acid here, this tRNA, if you notice, it enters into the E site, and the E site stands for exit. That tRNA now leaves the ribosome, leaving its amino acid behind. There is no longer an amino acid attached to this tRNA. Right? That was the green one there. And this is called an uncharged tRNA. And this one up here with its amino acid attached is called charged. Now this uncharged tRNA is going to go and find another enzyme to attach a new amino acid to it, another green one in this case, right? So another green one. And come back and carry out the process again the same way. Now this will continue until it reaches the end, the 3' end of the mRNA transcript, where there's a termination sequence. And that termination sequence will cause everything to kind of fall apart and release the protein, the primary sequence of the protein, to finish its folding.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site, as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
So this is how we ensure that the DNA sequence, which is transcribed into the RNA sequence, we ensure that we get the correct amino acid sequence. And so this is that DNA to RNA to protein. And by ensuring that we get the correct amino acid sequence, we now know that we're going to have the correct folding of the protein, because the folding of a protein is dependent on its primary sequence. So that brings us to the concept or the idea of regulation. Now, in genetic regulation, we have to ensure that genes are being expressed only when they should be. There's one thing you should understand about cells, no matter whether it's prokaryotic or eukaryotic, but that cells are incredibly efficient. They're like the most efficient systems on the planet. They do not waste anything, and they're not going to make something if they don't need it. We have lots of genetic regulation in our own cells, and bacteria are the same way. Now, to take a look at regulation, we're going to study something called the lac operon. And the lac operon is a set of 3 genes, LAC-Z, LAC-Y, and LAC-A. These 3 genes are all encoded or regulated by a single regulatory region. Now, there's one more gene called the LAC-I gene that is not within the operon. The LAC-I gene is a regulatory gene, and it encodes for a protein that is part of the regulatory system for the lac operon. So LAC-Z encodes for beta-galactosidase. Beta-galactosidase is going to break lactose into both glucose and galactose. Those are the 2 sugars that make up lactose. So we have glucose bound to galactose, and it will break this bond, thereby releasing the glucose into the environment for metabolism. The second gene is LAC-Y. LAC-Y encodes for beta-galactose permease. This is the pump that's found in the cell membrane, in the cytoplasmic membrane, that allows lactose to get pumped into the cell so that lactose will be available for metabolism. LAC-A encodes for beta-galactosidase transacetylase. This transfers acetyl groups. It's not necessarily required for lactose metabolism, but it is part of the lac operon pathway and plays a role in conversion of galactose into glucose. So upstream or away up here, this is kind of a little looping center here, but here's our genes right here. Here's the regulatory gene. This would be the LAC-I gene. And this LAC-I gene, LAC-I encodes for this regulatory protein that is going to move over, and it's going to sit on this regulatory region and prevent transcription. When lactose becomes present, it's going to attach to this regulatory protein and remove it. So it takes away the repressor. The repressor will be removed, and this will allow RNA polymerase to continue with its transcription. Now, regulation of the lac operon is actually dependent on glucose as well as lactose. It is a 2-step process, and it's called combinatorial control. So it's a combination of 2 things. Combinatorial. It's a combination of 2 processes that will determine whether or not the lac operon is actually produced to produce these 3 proteins. So this is why the lac operon is called an inducible operon. It is environmentally dependent. It totally depends on whether lactose or glucose and glucose are present in the cell. So I have a couple of different situations with this particular operon. If I have a high lactose concentration and a high glucose concentration, my gene is going to be off. If I have a low lactose concentration and a high glucose concentration, my gene will be off. If I have a low lactose concentration and a low glucose concentration, my gene might be on or off. We'll talk about that in a minute. If, of course, I have a high lactose environment but low glucose environment, my gene will be on. Okay, my operon will be on. And this all has to do with regulation. Okay? So I have 2 different things that happen. Number 1, I need to look at my lactose concentration. And in the lactose concentration, this is where my repressor becomes important. So let's take a look at diagram A. That's this one right here. Okay? I'm going to look here. This diagram's a little confusing. And in this diagram, I have my regulatory gene, which is my LAC-I gene. Think inhibitor. And this encodes for a protein called my lac repressor. My lac repressor sits on the regulatory region of the lac operon. And the regulatory region, the regulatory region is actually divided into two segments. Within that regulatory region, I have a promoter and I have an operator. Now, the operator, you can see here, the operator is where my repressor sits. And the operator works like a bouncer at a club. It decides who can go in and who cannot. And so this operator here is what starts as part of that initiation process in transcription. And RNA polymerase has to be able to move past the operator to start transcribing the gene. Okay? And the operator with this repressor on it is kind of like a bouncer there saying, hey, you know what? We don't need this right now. We don't need this right now. You don't need to make this, so go away. So no matter how activated RNA polymerase gets, which occurs at the promoter, no matter whether it's activated or not, if the repressor is sitting on the operator, RNA polymerase cannot move forward. So if the cell enters into a lactose-friendly environment, when lactose gets into the cell, then lactose will bind to this repressor. And when it does, they refer to this as allolactose, the inducer. When it does, the repressor is now bound by the lactose and it falls off of the operator and allows RNA polymerase to move forward and begin transcribing. So this is regulation by lactose. In order to remove the repressor off of that regulatory region, the operator region, I have to have lactose present in the cell. So lactose does get transferred into the cell. There is what we call some basal activity, basal transcription. Basal transcription means that there's very, very constant low-level transcription of a gene. And there is low-level transcription of the lac operon because we have to transcribe for that lactose transporter. So once lactose becomes available, enough lactose gets inside of the cell to tie up the repressor, and now my RNA polymerase is able to move forward. So this is the first level of control. But remember, this is a combinatorial control method. So there's a second level that's very important, and that's glucose. Now, in glucose metabolism, this is an indirect type of measurement. Let's write lactose up here so we know that's the operator is regulated by the absence or presence of lactose. But the promoter is regulated by the absence or presence of glucose. Now, when glucose is metabolized, when glucose metabolism is occurring, so let's write high glucose. When glucose metabolism is occurring, the cell needs something called cyclic AMP or CAMP. You probably, most students have heard of this in your anatomy course. And cyclic adenosine monophosphate or cyclic AMP, we call it CAMP. CAMP is used during glucose metabolism. So this means that CAMP concentrations are going to be very low during glucose metabolism. And in order for RNA polymerase to come together with the other enzymes responsible for initiation, I have to have cyclic AMP. So if my cyclic AMP levels are low, my RNA polymerase will not get activated. So regardless of whether or not lactose is present, if I have lactose present but I have a bunch of glucose still there, I'm not going to activate this operon because I don't have enough cyclic AMP. So high levels of glucose results in glucose metabolism. Glucose metabolism results in low cyclic AMP. Low levels of cyclic AMP prevents RNA polymerase activation. So I have to have a combination of high lactose and low glucose, this process right here. I have to have a combination of high lactose and low glucose in order for my gene to be turned on, in order for the operon to be induced.
Many bacteria grow on glucose before they utilize other compounds, such as lactose, as a growth substrate when both are present in the medium. Transcription of the lactose operon is controlled by two regulatory proteins. The catabolite activator protein can bind to the activator binding site and facilitate transcription, and the lac repressor protein can bind to the operator site and block transcription. The catabolite activator protein can bind to the activator binding site only if cyclic AMP is bound to it. When glucose is present, the concentration of cyclic AMP in the cell is low. RNA polymerase cannot bind efficiently to the promoter, and transcription does not occur unless the catabolite binding protein is bound to the activator binding site. Therefore, transcription of the lac operon does not occur in the presence of glucose. In addition, if there is no lactose in the medium, the lac repressor binds to the operator site, and this blocks transcription. When both glucose and lactose are present, there is insufficient cyclic AMP to bind to the catabolite activator protein, which therefore cannot bind to the activator binding site. Without the bound activator protein, RNA polymerase cannot bind to the promoter and initiate transcription, even though lactose binds to the repressor and prevents it from binding to the operator site. When neither glucose nor lactose is present, the concentration of cyclic AMP is high, and the catabolite activator protein is bound to the activator binding site. RNA polymerase can bind to the promoter, but it is blocked by the repressor on the operator site. When glucose is absent and lactose is present in the medium, cyclic AMP is present and binds to the catabolite activator protein, which in turn binds to the activator binding site and facilitates transcription. Lactose binds to the repressor and prevents it from binding to the operator site. RNA polymerase can now bind to the promoter and carry out transcription.
So this video, I did make it available in D2L in the course shell, and this video to me is just kind of the best video for understanding the lac operon and regulation of the lac operon. It explains both the operator site and the promoter site, the function of the lac-I gene and repressor protein, how we can turn off the repressor protein, we can activate RNA polymerase with using cyclic AMP, and how in different combination carbohydrate environments, we can see whether this gene is induced or not. So just for a quick review, these are some of the types of questions you should be reviewing for yourself on transcription, translation, and gene regulation. What's the difference between transcription and translation? So what is each process responsible for? How do we read the genetic code? Why is it often described as degenerate? Oh, I didn't. I'll explain that in a minute. What is the central dogma of biology? So know those different steps. What's the role of the ribosome in translation? And by this I mean you should be able to describe how a ribosome works, how it functions, the A site, the P site, the E site, what occurs in each of those sites. What's the role of RNA during translation? Would this be messenger RNA, transfer RNA, and ribosomal RNA? What are the functions of the 3 binding sites in a ribosome? And what is the Shine-Dalgarno sequence and why is it important? The Shine-Dalgarno sequence is going to become important in a second sense in mutations, just to give you kind of a hint. Real quick, I just want to talk about the genetic code as degenerate. I'm going to go back to an original slide here when we started, and I'm going to erase everything that's on here so that it's a little easier to see. And what we mean by a degenerate code, come on. What is meant by the degenerate code is that more than one codon will encode for more than one amino acid. So I have more than one codon for a single amino acid. For example, you can see here G, then C, and then in the third position, if you need to go back to my pen here if I can. when I'm recording, it doesn't like to switch the pens there. Okay, we'll go to blue. So if I go to G, this is position 1. C is position 2. And in position 3, I can have G, A, C, or U. So I can have G, C, A, G, C, C, G, C, G, G, C, G, C, U. All 4 of these codons will encode for the amino acid alanine. All 4 of them. And this is referred to as the degenerate code because in my DNA, alanine may be, I may have GCU, where that position is in my DNA, but someone else might have GCA. But this is the same protein, right, to carry out the same function, so it allows for some diversity, for some flexibility in the genetic code. And we need it to be flexible so that if a mutation occurs, and the 3rd position of a codon is most often where a mutation will occur, so that if mutations occur, it does not disrupt the production of those proteins. We have this again here for serine. Serine is, serine is encoded for by U, then C, then we have U, C, A, or G. So I actually have 4 different possible codons all encoding for the same amino acid. And again, this allows for flexibility in the genetic code. So those are kind of the review questions. I'll go back to that particular slide in case you want to pause the lecture on that so you can see these review questions. Use these to kind of work your way through the lecture and through the outline. Compare the notes and what I lectured on in here to the notes you may have from your textbook. And of course, there are some exercises in Connect that will help you to review this material as well. Hope you have a good day. The next lecture will be on mutations and repair mechanisms. See you soon.